the last thing too, because mine is done. Nothing like paperwork.
From Microbe TV, this is Q&A with A&V. I am Vincent Racaniello, and joining me tonight from New York, Amy Rosenfeld. Hello, Amy.
Hello, Vincent. How are you today?
I'm very well, thank you. And yourself?
I've had better days.
How long ago did you have a better day, would you say? Years, months?
No. It depends on what you define. This is a personal thing. So, like many years before the loons and everyone got sick.
Okay,
so that was
years.
more better days.
So when you
yes,
were young,
experimentally, probably, within the last month, I've had exciting days with our 414 and stuff. Yeah. But
so,
family-wise, before the lunatics all got sick and started not doing what they're supposed to do.
Well, hopefully it will get better, right? That's the thing. When things are bad, the good news is then they can get better. Right?
I don't know.
No, you don't
I'm
like
not
that?
going with that.
All right, let's start off the evening by asking everyone once again to support Amy's work on enteroviruses. Go to giving.
Yeah, and new enteros were just identified in Wuhan, and we're trying to get them. It's really exciting.
Giving.columbia.edu. Click Make a Gift. Type enterovirus. Your support of any kind would be lovely. Of course, it goes to Columbia, which is they're a nonprofit, so your contributions are certainly tax deductible there.
Right.
So,
I mean, Columbia is just the bank, is just the intermediate. The money goes to us, but we don't have our own bank account.
That's right. So, Amy, I wanted to tell you a funny story that I did tonight on the train. I'm not sure why, but I typed...
bank account?
I typed Vincent the Virologist into Google.
Okay.
And they found me. That was #1 hit, my Wikipedia page. So how about that? If you search for Vincent the Virologist, they find me. Apparently not too many Vincent virologists. And you know who was kind of stuck on with me? They said, people who searched for this also searched for Amy the Virologist.
And who did they get?
You.
Oh, really?
Yeah.
I have a Wikipedia page? No, I can't believe I have.
Oh, but you have a presence. If you search for Amy Rosenfeld, you will see things like your enterovirus.net and other things as well.
Well, most of the time you get some ESPN reporter.
Someone asked me to post the link to Fred Murphy's site. So let me do that. I'm going to share my screen here. So on Tweevo, this week, my pick was Foundations of Virology by Fred Murphy. So if you want to go to Tweevo, what is it, 76, you can click on that link, Foundations of Virology. And when you do that, here's what you get. It's very nice. Fred Murphy put this site up. It's at the University of Texas Medical Branch at Galveston, utmb.edu/virus images. And it's really a treasure. It is... he wrote a huge book. It's this ebook, Foundations of Virology, which is basically the history of virology. 600 and some pages, 2,000 images. History of virology. And then he also has on some of these other tabs here, he has electron micrographs. He has PowerPoint slides to go with the book. He has a chronological table of discoveries. He also has a link to his TWIV episode, and he has another link to information on yellow fever. I mean, this is he just puts this up for you guys to have. It's really remarkable. So if you haven't checked out his TWIV, you ought to do that because we don't want to play it. It's really cool. So check that out. Fred Murphy. Go to Tweevo or utmb.edu/virus images. That's very cool.
So somebody wants to know if Columbia takes a percentage, and no, they don't.
No, they don't. They don't take the VIG.
No
you know
percentage.
Do you know what the VIG, Amy, is?
No.
Oh, the vigorish.
No,
That's
I
what they
don't
used
know.
to call. Vigorish is what they used to call the interest. What the mafia would charge you, excessive interest rate. The vigorish.
I'm not Italian.
I happen to be first generation, by the way.
Yeah, I'm aware. I'm aware. I don't
Before
know
we
what I'm
get,
supposed to do with this information.
I'm just proud of my heritage. That's all. Before we get on to questions, I want to show you 2 pictures from the incubator. Here's the first
Don't
one.
do that.
What? Don't make
You
an
know.
accent. Okay,
Yeah,
from the
no
incubator.
accent.
So I had wall text printed, and I put it up today. So here's David Molesky's painting here, right? Ebola. And here is the wall text for it. And, as wall text goes, you can, it's got his name and birth date, the name of the painting, the year, the medium, and then a description of his influence. And then I thought it would be nice to get Fred Murphy's EM. And so I got a copy of that from Fred, and I printed it also. And you can see, man, this is the original 1976 EM, one of 45. And then I made a wall text for Fred as well. And let me zoom in on these so you can see them a little better. So here is Molesky's. He wrote this, actually. I asked him to send it. And determined to create a large-scale representation, he found Fred Murphy's images. With expressive strokes, he painted the outline. Okay, so that's cool. And then here is the Fred. So I got Fred's birth date wrong. I sent this to him today, and he said, Vincent, I'm not 99 years old. I was born in 1934. I don't know where I got this from. I thought I got it from his Wikipedia page. So I'm going to have to fix that, Amy. Anyway,
Yeah.
1976... electron micrograph 40,000x, and this is the story, which is just fascinating. And you can listen to, maybe I talked about this story on Tweevo, but basically they got some vials from an outbreak in Africa, and they arrived broken. And so Patricia Webb took a few drops of the fluid. She put it on cells. It killed the cells. She gave it to Fred, and he took. This is one of 45 EMs that he took. And this is the one that was the inspiration for Ebola. So, Amy, I think that's kind of a nice thing to have on the wall next to the painting, don't you?
Yeah, it's good.
And Fred is very happy, although he wasn't happy to be 99 years old, apparently.
Can't have everything.
I want to thank tonight's moderators. Tom is here. Les is here. I saw Vanity. I saw, who else did I see? Steph. I saw Steph. So thank you all for coming tonight. I appreciate it. We really need your help, and so it's great that you're here. Thank you so much. So Amy, why don't we see what's on people's minds? What do you think?
Sure.
I listened to the wonderful Twitter about the beginning of the virus at the meat market in China. That was so great. Thank you. No, it's not Twitter. It's Twiv. Yeah, that was Michael Warby, not Twitter. But that's okay. It's just a typo. No problem. Thank you very much for watching and for liking it. The incubator is powered down. Yeah, it is powered down. I shut things off when I leave. I'm at home on Wednesday nights because it would be too late to do it there. Yeah. So, Amy, I know you have a title of assistant professor. Do you teach classes or only do research?
Only research.
I wouldn't say only. I would say I do research because it's very important. Right?
Yes, I do research. It's very important, and I teach one-on-one because I teach Edmund.
She has trained some students and technicians over the years. She's a very good trainer, as you might gather.
Yeah, my students and techs have done well.
And who trained you, Amy?
You?
your parents, right?
Partly, yeah.
That was a leading question. I apologize for leading you.
It's okay.
Charlotte wants
It's
to know
always
what.
all about you, so it doesn't matter.
No, I hope I trained you well. I think I have because you're doing very nicely. It's not all
Yeah,
me.
hopefully I'll do well. We haven't moved on yet, so we need to move on.
What does Amy and Vincent think about what's going on in China with the outbreak? They're still the winners. They only had 6,000 people die. We had 1,000,000. What do you think of what's going on in China, Amy?
I just like the term they're still the winners.
Yes, I agree.
Um, and I think it's a travesty what's going on. Because they have these outbreaks, and then a lot of people don't have any immunity, and they end up in the hospital, and they end up dying and stuff.
They haven't vaccinated a lot of people, correct?
I don't know how many people they vaccinated. I don't think anybody knows. I don't know that anybody really knows that they've only had 6,000 deaths.
I think part of the problem is that their inactivated vaccines aren't faring so well. I saw an article saying that the protection against severe disease was declining, and if you give it an mRNA vaccine boost, it goes up again. But China will not accept mRNA vaccines. They have made a point of railing against them with incorrect
Really?
information. Yes.
Where'd you hear that?
I've read it in the newspapers, which of course could be wrong because I don't have any other connections. But
Yeah,
President
I
of
don't
China
know
has
that.
said,
I've never read that.
don't take the Western mRNA vaccines, and I think they should be getting boosted because that would help protect them. But it's not happening.
I don't know. There's conflicting data about their inactivated vaccine.
Yeah, there was a
And
study out
I
of
don't know
Brazil.
that. Yeah, well, I'm not sure that that's, I'm
Representative.
not sure.
Actually, it's
Yeah,
the
I'm
paper
not
you
sure.
sent. There's a study in Brazil where the protection against serious disease was declining, and they boosted with mRNA, and it went back up again.
Yeah, I know. It's just in that population.
Maybe we'll do it Friday on TWIV.
No, you already did SARS this week. Are
Oh,
you
we have to
going to do
do.
two?
No, I'm going to do that monoclonal paper that you wanted to do.
Oh, the really cool one with the hantavirus?
Yeah, I think that would be fun, don't you think?
I think so.
You approve?
Yeah.
Okay. Do we
Do we have
know
a
if
snippet?
the data show men being worse off for disease? All of my
That's
smart
Sabra
boosted.
Klein.
Yeah, so the men have about a twofold higher worse disease. Well, if you look at mortality, twofold higher risk overall. All men of all ages, which is probably concentrated in the higher age, I would guess.
Mm-hmm.
But yes, Sabra Klein, who was on TWIV, feels that estrogen, among other things, protects women, and it's an evolutionarily conserved protection because of reproduction. Anybody know how long you're infectious when you have influenza A? Well, it should be interesting that you should ask, because just on Monday in our virology course, I showed a graph of the duration of infectivity. Do you think I ought to show it, Amy? Would that be a cool thing to do? Yeah, I got it right here. I'll
Not
bring it up.
cool, but it answers the question.
It's actually a challenge study. Here it is. Let's see. Now I have to share my screen. Screen share. Okay, there we go. So on the left, this is a challenge study. And they inoculate these people on day 0, and they're measuring infectivity, virus shedding here, and it peaks on day 2, and then it looks like it goes out to day 8-ish. And this was a separate study where they did the same thing, and here it went out to day 6. So with that particular virus that they used, 6 to 10 days maybe. What do you think about that, Amy?
Sure, sounds reasonable.
But of course, it could vary with different isolates and so forth. So you never know.
Yeah, but it's on average.
Yeah. So this is Wednesday night of virology. It's become a tradition, Amy.
Oh, are you gonna sing Fiddler on the Roof or something?
No, but what is it? What is it that you said today? Hava Nagila?
No, that
Has
was
not,
yesterday.
that has nothing to do with it, right?
No, it doesn't. But there's a song called Tradition from.
Oh, yeah, that's from the Fiddler on the Roof, right?
Yeah,
Yeah.
it's really famous.
It's actually not a bad song, right?
I don't know. I never saw the play, but yeah.
All right, so a lot of people are talking about fourth doses. In the UK, we're on vaccine shot 4. Soon is this going on forevermore, quoth the Raven. Amy, is this going to go on forevermore?
I would hope not. I'm not clear that you actually need a fourth dose. I see no reason for one. And the data from Israel that described a fourth dose was very unconvincing. So I see no reason for this.
Amy, I have to say, and this is between me and you, okay? I can't let anybody else hear this. Last week,
I'm on the edge of my seat.
Daniel suggested last week that we would need yearly boosters. Oh my gosh, that I didn't say anything because he was in Panama. I figured, I don't know, maybe he got yellow fever.
Could be. Yeah, I don't know why you would need yearly boosters.
It doesn't make any sense. In fact, even Paul Offit doesn't think the first booster, the third shot was needed. But okay,
not
many
for
people
healthy
have
individuals. Not
but
for
the
healthy
fourth.
individuals.
But I think immunocompromised people, maybe you'd want a fourth because sometimes a fourth.
Well, it
helps.
depends on what your immunocompromised position is.
You're absolutely right. So the
but
paper
we don't seem to break it down. We just kind of lump them all together.
Anyway, I agree with Amy. We don't need a fourth dose. We don't need annual doses. This vaccine is working in vaccinated countries. It makes the disease milder. Sam says, what's the odds in-house of 4 vulnerable adults with a known work exposure that only one would show vague symptoms, and since smell is different, COVID didn't occur to me at the time? I don't understand what's going on here. Is Sam saying that 4 adults had known work exposure and one had vague symptoms and the other didn't? Because and no one got tested. This happens in households. Not everyone is infected, even if you have an exposure. Not everyone is a transmitter, so I'm not surprised at all. Are you surprised, Amy?
No.
So Rob wants to know, is it possible to have a biofilm that contains viruses, bacteria, and fungi?
I would assume so.
Yeah, biofilms can be quite complex. Absolutely. Mm-hmm. So, the answer is yes, Rob. That's about all we can say. If you were to remove every vaccine to ever exist, what are the chances Homo sapiens would still roam this planet today?
It's still roam.
Oh, we'd roam. A lot of people would have died. A lot of people would be crippled and still being crippled from polio, right?
For sure. And measles and smallpox and various other things. Yeah.
Yeah, be a lot of disease.
Yep.
German opera singer wants to know, do you think the COVID vaccine will need to be part of the infant vaccine schedule in the future, or will future generations just develop immunity by being infected multiple times with cold symptoms in infancy?
That'll be part of the vaccine regimen for small kids, yes.
Oh, you think so?
Yeah.
Do you think it will be a required vaccination?
Yes, at some point, but not for like 30 years.
Oh, you still think we're going to be vaccinating in 30 years?
Yes.
And so why would it be 30 years before that happens? I'm curious as to your thoughts about that.
What?
Before it's required for entry
into
school.
Because it took about 30 years for the polio vaccine to be required, about 30 years for the measles vaccine to be required, And all of their vaccines have not been given for 30 years, and they're not required.
Okay, that's a good point. I like that. And Miltz thinks this is the best thing on the internet, Amy.
That's great.
Thank you so much. This is very kind of you. And this is another question for Amy. Are there genetic variations in interferon that affects its ability to stop viral replication?
Yes.
There are a lot, right?
Yes. Casanova.
Yeah. So I think since this is a first line of defense, this is very important. And a lot of people have different efficiencies of interferon, and that can make a big difference in the outcome of an infection. But I don't think we know a whole lot about that. We know some, right? Like Casanova deficiencies in TLR3 sensing leading to
we just
herpes
know.
encephalitis, right?
Yeah, but that's not. I would say we're not even at the tip of the iceberg.
Yeah, I agree. I totally agree with you. We're not at the tip of the iceberg. Learning from you guys viruses that exist infect us humans has made anxiety worse in terms of viruses like EBV. Tuberculosis is not a virus.
Yeah, exactly.
That take a load on our most likely strained immune systems. I wouldn't say our immune systems are strained. I think they're highly functioning, and they protect you most of the time from continuous onslaughts of viruses.
Well, considering the fact that tuberculosis is a bacterium, like herpes virus is a bacterium, Okay.
What percent of preprints actually contribute to knowledge of COVID versus being a waste of time? Oh, you have to ask this of Amy Morris. Listen to what she tells you. What do you think, Amy? What percentage are a waste of time? Is it more than 50%? More than 50%? Now, you don't want to answer? It's fine if you don't want to answer. I will actually answer. I will say 50%.
I was going to say, yeah, it's definitely more than 50 plus 1. Yeah.
So many preprints actually never get published. They either are not submitted for whatever reason, or they're just rejected. And many of them are really not worth anything, unfortunately. But that's what we have. Non-science question. Tried to look for the full theme song from TWIV. Is there a link? Yeah. It's Ronald Jenkins. The one in the beginning, right? The one at the end is not Ronald Jenkins. But the one in the beginning is just a very short clip, right, from Ronald Jenkins. And the name of the song is Disorganized Fun. Let me see if this is it right here. No, that's not it. It's not Disorganized Fun. I have to look it up. It's a good question. It's not Disorganized Fun. But it was one of the Ron Jenkins, if you want to go through all his oeuvre. Got sick in December, PCR negative, flu and COVID, coughed up yellow-green mucus. Well, yellow-green mucus would mean a bacterial infection, right, Amy?
Sounds that way.
Some bacterium you got there.
Yep.
What's an entervirus, Amy?
It's an enterovirus, and it means it's a virus that replicates within the gut, the alimentary tract, although that's not really true anymore.
And
so it was viruses that replicated in the gut because you ate them.
And one of them is poliovirus, right?
Yeah. Polio, echoviruses, Coxsackie. Yeah.
And enterovirus D68, which is Amy's object of affection.
Yes.
Would that be a fair way to put it?
Yes. It is my baby. It's being cultivated.
Double vaxxed, had symptomatic COVID January. Any benefit of getting a booster?
No.
Nope. We could do a thumbs up, thumbs down kind of thing, right, Amy? No, you don't need a booster. You are in great shape. You are in great shape. Oh, look at Environmental says, hi, Amy, love.
Hello.
Isn't that nice?
It is. It's very nice. I like
I'm
the
not.
green heart.
Green Heart is lovely. Is that for St. Patty's Day?
I don't know.
Which was last week, right?
I don't know.
So Patricia, I actually call myself Earth's virology professor. That's different because I'm not Earth's virologist. No, Earth's virology professor, which, yeah, I like. Amy doesn't like it, but I kind of think it's cool.
I'm passing.
My
Okay.
MD said 3 months recovery and then vaccine after 3 months. Is she right? Yes, 3 months is what Daniel says also, correct?
Yeah.
Yeah. And look at, what's it? An enterovirus. Yes, she explained what, that's it, yeah. VNA, can't swing a dead cat these days without heating a preprint. And you asked this question before. We did. Amy is the rock star of virology. That's right.
No, it says both. You're both rock
Oh,
stars.
both. Sorry. I've spotted a curious thing in the Sanger Institute Genomics English data looking at my North London borough. Now 88% BA2. Okay, starting in February, that's one of two messages. Well, Alpha Delta are extinct, a low scatter of single cases of old lineage B, blah, blah, blah. Okay, yeah, but it's not surprising. The older ones, there's a little bit here and there. It may take a while for them to peter out. By the way, Amy, you remember at Ian Lipkin's thing last week?
Yeah.
What was the Vincent Price movie? Was that The Last Man on Earth?
Yeah.
So apparently that was based on a book called I Am Legend. And
Yeah.
3 movies were made, one with Vincent Price, one with Charlton Heston, and one with Will Smith, right?
Yes.
And Ian showed clips from all of them, right?
Yes.
Okay. I just want to know, maybe I need to watch them all.
Okay.
I Am Legend. I liked the Vincent Price one. I thought that was great from the few clips he showed.
Yeah, I mean, it was the best out of the 3 of them, although I do have to say that Dustin Hoffman was kind of funny.
You thought he was funny?
Yeah, in Outbreak, he was kind of a twit. You don't think he was kind of a twit?
He was a twit, but he was trying to be serious, right?
Yeah, but he was still a twit.
He was a twit.
He was a twit.
You're right. I agree with you. I totally agree. All right. Why is it so hard to develop dengue fever antiviral? Number of cases have increased. Why is that, Amy?
It's hard to develop an antiviral that doesn't have side effects, right? Because there are a lot of similarities between the pockets of and the active sites of polymerases and proteases with your host. So you don't necessarily want to kill the host as you kill the virus, right?
Yes, but I think you could make dengue antivirals. I don't know why. Maybe it's for lack of trying.
I don't think it's all that easy. I don't
No?
think it's all that easy. No.
All right.
I mean, it's not been easy to make Picorna antivirals.
No, that's true. It hasn't been.
So I don't know why you would say it should be easy against dengue. I don't think so.
I guess I'm spoiled by the recent spate of antivirals against a variety of viruses. I mean, we have over 30 for HIV. We have a bunch for Hep C.
Yeah, but
right?
they're not really. So outside of Hep C,
Yeah.
you have to have 30 for HIV because there's a huge amount of resistance.
Yeah.
So I don't know why you would think that. So right then and there, that undercuts your argument that it's been
Okay. So Pete, this is his final question. Are these reintroductions from the Far East? Who knows where they come from? I don't think you have any way of knowing. But we know Delta lasted longer in China than in other places, so it's possible, yeah, because travel can spread virus, yeah.
Really? Delta lasted longer in China?
Yeah, when Omicron was dominant elsewhere, it hadn't yet become dominant in China, and Delta was still circulating.
Well, I don't know that. I don't know. I don't know that I would have interpreted it that way, but okay.
If you do Vincent the virologist, you also get Vincent Munster. Yes, that's good. But I mean, he doesn't do what I do, right? he's a working scientist and doesn't do social media stuff, so that's why there's a difference. But yeah, I don't. There probably are other Vincent the flavivirus guy that Dixon knows. Vincent Dubé or Dugay or something like that. That's another
I don't know.
Vincent.
I don't know who Dixon knows.
What's the difference between EV68 and 71?
Huge! Huge! Huge!
Start telling us.
Well, so 71 is orally fecally transmitted. It's a gut virus, and then it disseminates into secondary sites of infection, which generally had been correlating with blisters in the mouth and feet and hands. Whereas 68 is a respiratory virus. It replicates within the respiratory tract. It's very sensitive to acid, so it falls apart if you were to put it in the stomach. And it does not correlate with a rash and blisters on your feet and hands and mouth. And it correlates with severe respiratory distress syndrome sometimes if you have asthma and stuff. It uses a completely different receptor. It's unknown what the receptor is, whereas EV71 can use SCARB2 or PLG, whatever it is. And if you have persistent lung infection, you can get it to adapt to heparin sulfate. And the structure of the Variant is hugely different. Like, it's much smoother than 68. So huge
Amy,
differences.
which one do you like better? Which one do you like better?
I like them both, but 68 really is cuter. Okay. It's more interesting.
You have a nice painting of 68, don't you?
I do. I have a lovely painting of 68. But it's also more interesting.
Is it behind you?
It's next to the door. It's also more interesting because it's not clear how respiratory pathogens without a viremic phase that stay outside the body would then cause a neuropathology. And so while it's not clear how 71 disseminates, it follows a more traditional path in the fact that it was a gut pathogen. Although I do have to say, it first was identified, and then in Bulgaria in the 70s, there was a huge outbreak of polio-like symptoms that was not polio, but 71. And then it just correlated with rashes, and now it somewhat correlates with AFM. But like the whole idea of what happened in the 70s, where it made the polio-like symptoms is like really interesting. And unfortunately, we can't find that virus. So.
90-year grandfather asked for advice on the fourth dose. In your professional opinion, should I tell him it's unnecessary in his age group? My opinion would be that 90 years is immunosuppressed. You should get a boost. You should get a fourth dose. What do you think, Amy?
I don't know. I don't know enough about him.
Okay.
He might not be immune suppressed. He might actually have a pretty robust immune
it's possible.
system.
But
I mean, my grandmother was 100, and she had a really robust immune system.
I know,
that wasn't why she died.
Without data, that's what I would say.
Yeah.
Scary article in Forbes on brain shrinkage after infection. What would be your hypothesis on how COVID causes brain changes without infecting the brain? Do other viruses do that?
Hypoxia.
Mm-hmm. And so that must be present even in mild COVID, right? Because a lot of these cases had mild COVID.
Yeah. When you can't bring in enough oxygen, all of your blood, it has low oxygen. There's gradations of hypoxia.
Yeah. I remember on Twitter you were talking about an experiment you used playing cards in virus transmission. Where can I find more information? Who did that experiment? Was that the guy at the Common Cold Unit in the UK? What was David Tyrrell?
I think so.
So these are papers on rhinoviruses, and I think the PI is David Tyrrell, Tyrrell, or some combination of R's and L's. So you could look it up. Rhinovirus challenge or transmission experiments. And these are from the 60s, I think, Amy, right?
Yeah, for
They're
sure.
quite old.
For sure.
A friend of mine is an artist. She paints on hubcaps. She's painting a virus for me to send to you. Well, that's cool.
Yeah, very cool.
Maybe Amy could put it on her car. I think that would be sacrilegious, right?
Well, I have no car. So
Maybe
I don't
you
know.
have
I
one
don't know.
one day.
I don't know what car. I don't know what car we're putting it on. My invisible car.
Yeah, well, a hubcap would be a great thing to put on the wall. That's cool. How bad the COVID death statistics are in Russia? Are they accurate? I have no idea. No idea. What about you, Amy? Do you have any idea?
No.
I really don't know. I'm sorry. Is your wiki page correct, Vincent? Do you need to fix anything?
Since I take care of it, it should be correct.
The colors on the Ebola paintings make me think of cave paintings. Yeah, that's an interesting point. Yeah, the palette is cavey-like, isn't it? Yeah. Mm-hmm. All right. What do you think about the 25 microgram dose versus 10 between Mrna and Pfizer for kids? What do you think, Amy?
I think it's fine.
You think it makes a difference in immune responses?
Well, clearly.
Somebody asked that of Daniel last week also.
And what did he say?
Didn't think it mattered.
He's wrong.
Yeah. Everyone's wrong sometime, right?
I don't know.
If COVID vaccine is receiving more scrutiny than others in the past, do we assume other vaccines might produce the same type of findings that question the safety of such vaccines post-release? Now, I don't know what you're talking about, question the safety of. We're not questioning the safety of any of these, and no, they're all safe.
I don't think that they receive more scrutiny than others in the past. I don't think you can actually make that comparison. So I don't know what the question is.
If brain shrinkage happens after every infection, wouldn't really become an issue in 5 to 10 years as we cycle asymptomatic infections annually.
Well, I mean, you're not always hypoxic, right?
No, not every virus makes you hypoxic, that's for sure.
Right. And there's degrees of hypoxia and there's degrees of damage. And it's like there's degrees of dehydration where you can rehydrate, and then at some point you get to a point where you can't rehydrate, but most of the time you lose water and then you rehydrate. And you would know that if you got too dehydrated because it would be really painful.
Do you think polio causes any hypoxia?
No.
Even if it is affecting respiratory muscles?
No.
Amy, thanks for the excellent input. I love the way you don't take any crap.
Oh, okay.
Do you view yourself as not taking crap, Amy?
No.
No, you don't. That's just the way you are, right?
Yeah, I mean, that would imply like there's a conscious thought. No, I don't have the conscious thought. It also may imply that I'm manipulative, and I'm not manipulative.
No, I don't think that implies it, but you're certainly not manipulative. All right. Matri wants to know, boosters, if fully vaccinated, comorbidity groups are still vulnerable to bad outcomes, is that different than the argument made last year that only those with comorbidities need to be vaccinated?
No. It's the same argument.
Everyone needs to be vaccinated, no?
Yes.
Are defective particles primarily the product of defects in the bits and pieces or of assembly of those pieces? A complex virus more likely to make more defective particles than simple ones?
Well, I mean, let's look at it. Herpes and DNA viruses are some of the most complex particles, and they have fewer defective interfering particles than like polio does, because there's DNA repair, recombination, and replication enzymes in the cell that correct the differences, right?
Yeah. So it's a mixture of physical defects and genetic defects, but small viruses have very bad production quality as well as big viruses. So I don't think size matters in that sense, right?
Right. Right.
Vincent, how am I going to cut grass now or garden with deer camping out in the backyard? Remember, I was talking about how I cut the grass and I
I know,
aerosolize.
but you've been doing this for 35 years. You're going to continue to do it for 35 more.
Yeah, so Gwen, I think the risk is very low. That's the point.
I don't think that there is a risk.
And there maybe not, because maybe it just doesn't transmit to people, and I've done the experiment, right?
Well,
So-
the other thing is you're not eating anything.
Yeah, maybe eating
You're
is
inhaling
important.
it, so it's a completely different set of cells.
Yeah.
It's a different
Yep.
tropism.
Could be.
So I don't know what the point is.
Well, she's just afraid.
Yeah,
that's
I understand.
all.
I
So
understand.
that's okay. We can assuage fear. It's fine, right?
Yeah.
So anyway, Gwen, go cut the grass. It's therapeutic.
Really?
Yeah, because nobody's talking to you. You're just out there with your own thoughts. I really like that.
Oh,
And you know,
don't
when I
I've
cut the
never
grass,
cut
I
the
think
grass.
of podcasts to do. I think of papers. I think of hypotheses, and I think of stuff. It's really good. It's a really good
Okay.
time.
Okay. Well, maybe
I've-
you should start cutting the grass all year round.
No, I don't need to do it in the winter. It doesn't grow in the winter. Can you imagine if I was out cutting the grass every week in the winter? They would send the police,
Why would they send the police?
because they would say this guy is out of his mind.
I didn't say you should do it naked. So
No,
I'm
just
not
cutting the grass when there's
okay,
nothing to
I'm
cut.
not clear. Okay,
I
whatever.
found you guys
was
a
just
year ago
pathetic.
when I was desperately trying to learn about COVID. I haven't missed a Q&A, Twiv, or update with Daniel. Oh, that's lovely. Thank you so much for being a loyal listener. It's great, and I think you're learning a lot, right?
Yeah, for sure.
My wife is from Beijing. Tried to keep COVID out and focused on vaccinating children. Older people tend to be suspicious of vaccines, so now their most at-risk people are at huge risk. Yeah, that's unfortunate. Same thing in Hong Kong, right, where the older
Yep.
people don't want to be vaccinated. Now they're dying. Yeah.
Yep. All right, it's 9:12, so like 3 more, and then I have to go.
I've seen news articles of the vaccine helping reduce symptoms of long COVID in people. Could getting another vaccine serve as a treatment option?
Sure. It's not
Yeah,
going to
so-
hurt you. So, sure.
But it's only a 30%. Daniel has said it's about 30% of long COVID people see an improvement in some symptoms, not everything. So I don't know if it's the answer, but it's better than nothing.
Well,
right?
30% is, yeah, I was going to say 30% is pretty high.
Any
And
news
I
on
don't
that?
see why it's any different than like when our favorite polio person said we should all get another dose of OPV.
I can imagine who that is.
Yeah.
Any news on the vaccine being created by Walter Reed? This is the protein based
With
vaccine.
the ferritin thingy?
Yes, that's right. You may have got a lot
I
of
don't
press
know
a few
anything
months
about
ago.
it.
I don't know what's happening. Yeah, I mean your guess is as good as mine. My wife and I saw our physician. We told her about Paul Offit's stance on boosters. She told us to get the booster. We have 2 Pfizer jabs. Confused what to do.
Depends. Paul Offit is very clear. He says if you don't have comorbidities and then if you're below a certain age, you don't need a booster. If you're above a certain age and you have comorbidities, he thinks a booster is fine. He's very clear. Read his editorial.
Philadelphia Inquirer. Linked in a recent Twiv.
Mm-hmm.
Yeah.
He's very, very clear.
Don't a very large percentage of Americans have comorbidities, and thus shouldn't boosters be recommended for a large portion of the U.S. population?
I have no idea.
I don't know if we have we certainly have a large fraction of obese people, but um, is that the main, what is the main comorbidity, Amy? Diabetes? I thought it was diabetes, obesity.
I don't know. I mean, I don't know. I don't know. And I also don't know if when you're talking about diabetes, if you're talking about like type 1 diabetes, which your beta cells get destroyed by some unknown mechanism, or if you're talking about like insulin resistant diabetes and type 2, because, yeah, I don't know what you're talking about. So I don't know. I mean, it's not so clear cut.
With Sete and Crotty, new study on COVID vaccines. Did Amy say you think? Did you say you think?
Yes. If it's the paper, I think that was on bioarchives about the 4 vaccines, yes.
What did they say?
I sent it to you and Daniel this morning. You guys can discuss it tomorrow.
Okay. A mainstream news source says BA2 is 20 times more contagious than the original Omicron, but not any more deadly. I've learned from you guys to be skeptical of claims. Wanted to get your take on this.
Well, if it were 20 times more susceptible, it would be the predominant variant, right? Months ago. I don't know how they're defining contagiousness without like some data. So I don't really know what this means. I don't really think we should discuss these kind of questions, because I think actually these statements should never be said. They should be edited out of newscasts.
Yeah, this is completely misuse of the word contagious. Okay, it's just, we don't know anything about why BA has some advantage in some areas.
Exactly.
Your thoughts on the Baylor vaccine now being administered in India? Is it as effective? Seems it will be faster to produce in low-income regions.
I think that's why he did it. It's faster and it's cheaper.
Yes, but we don't know how good it will be against severe disease. They didn't test it against disease. All they did was said, when we give this to people, they make a certain level of neutralizing antibodies. That's it. No T cells checked, no disease. So we're going to see if it works.
Yep.
So it's a toss-up between getting it fast to people and what if it doesn't work? What's good is it to get it fast to people if it doesn't work?
Oh, we don't really need to focus on the details.
Shot 4 in the UK is for immunocompromised and over 75s. Okay. Thank you, Ian, for your contribution. In New Zealand, we sequenced all cases to back up epidemiology. If we make mistakes during replication all the time, how is a single genomic sequence arrived at? That's Amy's answer. How do you get one consensus out of a sequence?
So if you're doing Sanger, it's the predominant genome that is in the population that you pick up. You don't even see the others. And if you're doing next generation, it's an average. It's what is 50% plus 1, basically.
Yeah.
It's the easiest way to think of it.
So you never get the minority variance, right?
Well, you get the minority variance if you do next generation sequencing, but not Sanger sequencing.
So when they get isolates from patients and they do whole genome sequence, is that next generation?
Today it is, yeah.
So they have to make a call at every base position, right?
The sequencer makes a call, and then what you do is you say, I have this read is, if a read is less than, let's say your cutoff is 1%, it's considered garbage. So it goes into the trash can by the sequencer. And then you piece together and you say, okay, so 50%, let's say it's simple, 50% plus 1 has an A, so you call it an A. And then you make a list of the variants that you see that have like C's and G's and T's at that position. And you can do that by percentage.
So basically what's given to the repositories is a consensus. But you could go to the original NGS data and find all those variants, right?
Yes, and that's what people did in the beginning to find all the variants at the beginning that Theodora talked about at
Yeah.
one point. She
Right.
says they're all present, but they're at a very low percentage. Yeah. So sometimes it depends on what percentage it is, you miss it. So that's like what Patrick does. Is he goes through the garbage bin and he says, well, maybe we shouldn't throw these out because yes, I agree that they're at a low percentage, and that would suggest that it's a sequencing error, but maybe not. That's what he does.
Good. All right.
Basically.
What do we know about the bird flu outbreaks I see all over the news? They're culling chickens and ducks. Not chilling, they're culling them. Yes, well, there are periodically outbreaks of avian influenza in commercial farms like this. It just happens periodically. And they respond by killing the chickens to stop the outbreak, yeah.
Well, there's no antiviral for chicken.
No.
Yes. All right, one more and then I have to go.
If mRNA vaccines are supposed to be quick to change and plug and play, why are the boosters the same as the original shots?
Because you'd have to go through a trial, a modified trial to test to make sure that whatever you've changed works and gives you the same response. And since there's no need to make a variant specific one, because the original sequence that we've been using generates a wide variety of antibodies, then you don't need to change.
Yep. All right, Amy, thank you very much.
All right, I forgot what we're doing tomorrow, but I'm sure we're doing something. I don't know exactly.
Thank you, Amy.
All right, have a good night.
Good night.
Bye.
Bye-bye. You expect this virus to get weaker over time, yet more transmissible. No, I don't on either count. So let me riff a bit on transmission again. We haven't done this for a while because I think about this all the time, and I have not been cutting my grass, but I still think about it. Transmission is the movement of a virus from person to person through a population. And we can talk about increased transmission, which could be a consequence of people going to a football game, thousands of people together, or the virus might change. And unless you do experiments in the lab, you can't tell if the virus has changed or not. So you can't tell if the virus has become more transmissible, which means there's some change in one of its properties that allows it to transmit better, okay? I mean, this is a very nuanced thing that nobody is going to pick up unless you listen to these live streams. Even the virologists are not going to get it. They don't think about it. But it's not that hard. So we talk about transmission of viruses, but transmissibility of the virus is its intrinsic property, whereas transmission is the sum of that plus all the human behavior and immunity and so forth. So transmission can be impacted by immunity. If a population is immune, it will impact transmission. So a virus that evades immunity will transmit better in a population. It may not become inherently more transmissible, but it will transmit better because it goes into an immune host and it can still replicate, right? So it will establish infections. So I do think that, all the variants, in my opinion, are the result of immune selection for viruses that evade antibodies to some extent. And as a consequence, they have transmitted better. And this is quite clear for Omicron, where there's no intrinsic change in the transmissibility of the virus. There's just its ability to evade antibodies, and therefore it establishes itself better in an immune population. So given that definition, okay, yes, I think as this virus reproduces in immune populations, variants will continue to be selected. I'm not worried about it. So in an immune population, yes, the viruses will infect people. However, they will not cause severe disease for the most part, unless you're extremely old and immunocompromised, certain types of compromised disease. So that's what we see with the common cold coronaviruses. They change every so many years. They evade immunity. You get infected, but you barely get sick. So will this virus be the same? I think the data are telling us that it will, because we see that in vaccinated or recovered people, the disease is generally mild. It's not always mild. I know some people die, but that's the way it works. And if you're 85 to 90% protected against severe disease, hospitalization, and death, that's a damn good vaccine. You're not going to do much better than that. What boosting every year would do, and it's not recommended for everyone anyway, but what it would do is prevent you from being infected for 6 months. But after those 6 months, when your antibody levels go down, you're going to get infected, but you're going to get mild disease. So I don't think the virus is going to get weaker. I think it will be transmitting better in a population because of immune evasion, just like influenza virus does every year. Okay, think influenza, variants arise every year that move very quickly through a previously immune population because they evade the immunity. Yep. I think you and Daniel also disagreed about testing. Yes, I do disagree with Daniel on testing, and he always makes that statement. I don't remember what it is. Test more, right? You guys know what it is because you listen to him every week. I listen to him too, but I can't remember what his statement is about testing. So he just wants to test everyone. I don't think it's needed. We don't test everyone for influenza. Now, when do you need to test someone? If someone has symptoms and then you want to know if you have to treat them or not, may have a comorbidity, they may be older, whatever. Yeah, you have to test them. And that data can go into the epidemiological profiling that we need to take track of variants. We don't need to test every student in my Columbia University virology class every other week. No, especially if they have no symptoms. That's not necessary. It's not getting you anywhere. All it's going to do is say, oh, go to your dorm room for 2 weeks, and that's not necessary. That's what I'm talking about. And I'll bet if he and I talked about it that way, we'd agree. But I have had disagreements with Daniel, but we have reasonable discussions, and we agree to disagree. I think it's fine. The problem I have is when people say you're wrong. All the data says this, you must be wrong. Why don't you just have a discussion with me? That's the way to do it. So if you're 90 or over, get a 4th dose. It's fine. I think it makes a lot of sense because you're likely to be immunocompromised. Not guaranteed, but likely to be. If a new variant or strain of corona emerges in the future, which corona? Would our immunity, must be SARS-CoV-2, prevent pandemic? Why didn't previous common cold coronavirus not give immunity to SARS-CoV-2? Because they're too different, right? They're too different. They're antigenically too different. The antibodies that are made do not give you protection against SARS-CoV-2. So if SARS-CoV-3 comes in 10 years, which it might, you're not going to be protected. Well, who knows? Maybe it'll be more similar to SARS-CoV-2 than the common cold coronas, right? Maybe all of us who have been vaccinated and infected and recovered will be partially. It depends how different it is from SARS-CoV-2. But frankly, if it's very similar, it's not going to cause a pandemic, right? It would have to be very different. And that's the definition of a pandemic. Most of the world gets infected, or many countries get infected, and that can only happen when there's very little immunity. So it would have to be a very different virus, yeah. My mother's 88, had COVID in 2020, is triple vaxxed. So I don't know anything about your mother's health. If she's healthy, man, she has had infection, she's been triple vaxxed. Seems to me you don't need a 4th dose. If you tell me she has B cell deficiency, then yes, of course. But sounds like not. Well, I disagree with Osterholm on this point. I don't think a 4th dose is a good idea. Why does he think it's a good idea? He probably doesn't explain it. And I don't give a damn about his podcast because he would never come on mine. And then when COVID came, I think I should make a podcast and get famous. I'm sorry. I don't like that kind of behavior. But that aside, you don't need a fourth dose for everybody. He thinks I don't care what he thinks. I want to know why. We don't think it's needed for most people because you don't get serious disease with 3 doses, right? Inactivated versus mRNA question. What do you want to know? You want to know what the difference is or which is better? Please re-ask the question because it can go 2 different ways. I'll take that as meaning we wouldn't have magically morphed into a virus-free super being. No, of course not. Viruses have been here since humanity arose and before. Viruses were here billions of years ago. So if we had no vaccines, we would all be more infected than we are now. Yeah, for sure. Is there any chance that any of the known prion diseases could progress to epidemic level in humans? Well, I mean, the mad cow disease in humans was an epidemic, right? We had cows getting mad cow, and then we didn't pick it up for a while, and then the cows got in the food chain. We ate them, and cooking. You can cook your hamburger for 2 hours until it's charred, and you'll still get prion disease because you can't inactivate it with heat. All that prion went into the food chain and many hundreds of people got sick. So the biggest scare is, yes, if it gets into the food chain undetected and millions of people eat contaminated food, then yes, we have an epidemic. So that's why people freak out about chronic wasting disease, right? Apparently new prion disease. I'm sure it's been around a while, but we've just discovered it. We don't want that to get into the food chain. Here's the thing about cows and mad cow disease, bovine spongiform encephalopathy. It takes time for a cow to show the symptoms of a BSE. Most cows that are processed for meat are processed. They're killed before the time. They're killed within 2 years. They're fully grown in 2 years. They're killed, and they go into the food chain. That's too soon to see the symptoms of BSE. This is the issue. Now, in some countries, they check every cow for BSE. What do you think they do in the U.S.? Nope. Don't check every cow. Not doing it. They make a decision, risk versus benefit. Actually, I'm okay with that. I don't eat meat anyway. But in the old days, I did, so I could still develop a prion disease, I suppose. But I think the risk is very low at this point. That's a good question. I like that. There is a dengue vaccine, Dengvaxia. It is licensed in Brazil, I believe, and Philippines, and perhaps some other countries. Has some issues because it had bad reactogenicity in young children, and it was a consequence of one of the 4 serotypes in the vaccine. So it's not ideal. It can be used safely, but there are restrictions. Another one is being developed at the NIH, which I think will turn out to be safer. And yes, there are malaria vaccines as well. Several malaria vaccines, yep. Popular influential figures. I don't know what a popular influential figure is. This is a pundit who doesn't know anything, right? Argued last year the only people with comorbs should get vaccinated. Well, I never bought that anyway. Okay, so can you distinguish that from the view that people don't need a third? You get 2 doses, you're vaccinated. There's no comparison of the two. I think Amy addressed this before. No. With respect to the Epstein-Barr MS study, if there was an EB vaccine, would the vaccine prevent MS since vaccines don't prevent infection? This is a great question. It's a really good question. So, so what happens with the EBV-MS connection is you get infected with EBV. Everybody does, and then in a certain fraction of people, well, you all make antibodies to many viral proteins, and one viral protein, a certain fraction of people, those antibodies cross-react with a neuronal protein, and that's giving rise to the MS. That's the idea. So it depends on how much immunity you get from that infection, right? I don't know the extent of it. It's an interesting question, and I can't extrapolate from other viruses, right? So you get an EBV vaccine, and after 6 months, you now have memory, but you don't have very high serum antibodies. And so then you encounter EBV, and this you would have to get very young age. You have to be childhood, right? Which is fine. And then you get infected with EBV, and it takes a couple days for your memory B cells to wake up and make antibodies. In the meantime, EBV is replicating. So the question is, in those 2 to 3 days, do you make enough antibodies to that EBV protein to be a risk? And I don't think anyone knows the answer to that. It's some sort of thing that we'd have to make a vaccine and observe if a certain fraction of MS goes down. So that would be a long-term study, right? Really good question. Really good. Like that one. I should put that in the bloody... I want to put that in the... how can I copy this comment? I want to put it in the... Oh, it's going to be immortalized in the video. I use that in my class. It's a good question. Interferon and ribavirin, what percentage cured my hep C in 99, but my daughter is convinced I got an autoimmune from that. I don't think you would get an autoimmune disease from interferon and ribavirin. That's what was the standard of care for hep C before the direct-acting antivirals, right? And it didn't work in everyone because not everyone responds to interferon because of genetic differences that we actually alluded to earlier in this, right? As A&V will manage global public health during the next pandemic, what will you do differently? Well, we won't, but I think it's an interesting hypothetical, right? Unfortunately, A is not here, but so the key is rapid response, and I think we're kind of hamstrung, right? Because this virus emerged in Wuhan, clearly in the epicenter was the market, the Huanan market, and on the 27th of December, physicians in Wuhan recognized that this was a new virus causing pneumonia, and it was contagious. It spread from person to person. And that day, they should have put up the cordon sanitaire, but they didn't. They waited for a month. The CDC waited a month, and in that month, the virus escaped. So what could we do about that? We can't do anything because we're dependent on them to tell us what's going on. You know, it's not even clear to me that they would have released the genome sequence if someone hadn't done it for them, right? Jeez. So, all right, so that's out of our hands. But what could we do here? Well, we took a long time to start testing, to start telling people not to travel, to start quarantining, to start restricting travel. All those things need to be done immediately based on where we see the first cases. We have to develop test kits very quickly. And, you know, the two viruses that are going to do this are flu and corona. I want to have, in the meantime, broadly acting antivirals and potentially broadly acting vaccines for both developed that will be needed and stockpiled for the deployment. A lot of things you can do. I'm not doing it. Nobody's going to ask me. And that's fine because I get to stay here and talk to you every week. And that is why that is why this will continue every week because there are all these interesting questions to deal with, and viruses are always an issue. Why no tuberculosis vaccine? Well, there is a vaccine, BCG, but it's not very good, right? We could be better. I think people are certainly working on it. Are there any known gravity-dependent processes involved in viral reproduction? That's a good question. So there is an astronaut who is a virologist. Did you know that? Let me pull up this cool thing here. All right, we're going to get rid of Keynote. Oh, how I love teaching you guys. Let's see, Twiv, Kate Rubens. Oh, I love telling you these things. So here she is. All right, I have to share my screen. Now, if I had a producer here, I could just say, hey, producer, share that clip. Here she is, Twiv, what is it, 682. It's actually the second time she was on Twiv. Kate Rubens. So she's a virologist, and she does experiments in space. I also had her on another time. Where is the second one? Here's another one. That's 682, but we had an earlier one where we interviewed her. I don't want to go to the YouTube. No, there's no picture here. Maybe no, there was video of that. Anyway, she was. We had her at an ASM meeting. So she could do that kind of experiment. She's growing cells in space, but she doesn't go up all that often, right? But she may go to Mars, and then she could do a long-term experiment. But so far, they're not doing virus infections in space, right? They have to get the cell culture going and make sure all the protocols for it to be safe. I don't think there's any gravity-dependent processes, to make a long story short. But certainly, we should look at it, right? Cells can grow in space without any problems. Has interest in virology degrees increased significantly due to COVID? I would say no. I think there aren't that many programs in virology in the country, right? To get a degree in virology is pretty rare. We don't offer one at Columbia. We offer a degree in microbiology and immunology, right? There are some virology programs. Harvard has a program in virology. Baylor has a program in virology where you can get a degree in virology, but there are not that many. But I think the interest has been on the part of researchers who are in other fields, and they jump into COVID, and they want to do COVID, and most of them want to do it because they can get money. They think they can get grant money, and I think that's a crappy reason to do it. So I've had a lot of interest in my class, right? But my class is always big. I always have 130 students, and it didn't really go up. I added a summer session. Maybe that helps too, but I think I've had more interest from you guys in virology than I see at Columbia. So I think the interest is in certain areas only. What determines the replicability boundary? Minimum viable number of base pairs. You mean the minimum number of base pairs of the genome? Well, I don't think it's the minimum number. It's the number of genes that you need to reproduce the genome and make infectious particles, right? And some proteins are dispensable. So you can remove ORF8 and the virus reproduces. But what other genes can you take out? We don't know for this particular virus. I'm sure some of them are dispensable. There are many, for example, immune evasion genes that you don't need in cell culture. You would need them in a host, of course. But I think it's all about the proteins that are needed, not necessarily the length of the genome. I remember in one of the first Q&A, you answered a question about the media friends. You said they have to sell newspapers. That's engraved in my mind forever. It permeates me. That's what I've been thinking that for years. They have to sell. The press is conflicted, right? Whatever you watch, CNN or MSNBC or Fox, they're all conflicted because they have to sell their product. They have to get you to watch. And they do that. They get your attention. They make sensational sayings. And I don't. I just tell you the facts. But yeah, I'm glad it's engraved in you. I'm glad that certain things stick, because I think that one is a really important one. It seems like to some people there will never be an appropriate time to relax precautions even after vaccinated. Now they are back to, do we need forever mask mandates? No, I don't think we need mask mandates. As soon as the young kids can get vaccinated, and they're not all going to get, because their parents are not going to vaccinate them. Not all of them. Some will, of course, and that's great, but many won't. As soon as they can be vaccinated, then we don't need mask mandates. They'll end on public transportation, and it'll be up to a business if they want to require you to wear masks. Already in New York, the streets are 20% people masked. My class is in Columbia. Mask mandates were raised last week. 20% of the class wears masks. The rest don't. I don't wear a mask. I love lecturing without a mask. I want to go back to normal life. I think I can assess the risk and say, yeah, if you're these vaccines work really well. They're not perfect. I'm willing to take the chance. I know the odds. I'm willing to take the chance to go out into life. And so I think it's crazy to think that there will never be an appropriate time. No, of course there will be. There was after the 1918 flu, for God's sakes. We went back to normal. That was more horrible than this pandemic. And we had nothing to stop it. And it ended, and we went back to normal life. So it is just not correct to say we're not going to be able to do that. I have chronic lymphocytic leukemia and am obese. I just made plans with my GP to put me on antivirals and monoclonal if I should contract COVID. How important is planning ahead? This is perfect. You're doing exactly the right thing. So, yes, you are immunosuppressed. You are have a comorbidity risk. So, even if you're fully vaxxed and boosted, you might still have a bad outcome if you get infected. So you should be given antivirals or monoclonals. They will save your life. And so, yes, have a conversation, everyone who are in similar positions, with your physician, and say, I want this if I'm testing positive, and make sure it happens. Absolutely right. That's a great point. My husband got hand, foot, and mouth from our grandson. It was the worst thing he had suffered in 62 years. Awful. Coxsackie, why him and not me? Because. So here's the thing. Usually you get Coxsackie as a young kid, and you get some immunity, so then subsequent infections are not as severe. So kids typically get hand, foot, and mouth. So your husband has probably some unknown compromise in his immune system that either made his response to his initial Coxsackie infection poor, or compromised his memory, whatever. He has some issue with immunity for sure, and that's why this happened to him, because most of the time it doesn't happen to adults. Could Daniel post the steps to medicating a patient should take if tested positive? He does every week. He goes through the recommended treatments every week, and if you just go to one of the pages here, let me bring it up for you. Okay, let's go to Microbe TV, and then you go to the latest clinical update. Here we have the last one. Let me. Producer, can you share my screen? Thank you so much. I don't know. Maybe it's just always going to be me doing this. Anyway, so you go down here. Anyway, you listen to this, and I don't usually put the links for this here, but he talks about the recommended treatment every week. Okay, every week he does that, and you can glean. There's a website that he uses to recommend that. So listen to one of them. If you don't, just one. It's 30 to 40 minutes. Will there be a SARS-CoV-3? Yes, I'm sure there will be, right? There was SARS-1. They could have called MERS. Well, MERS was different enough. It was not a SARS-like virus, right? It's more distantly related. So, but SARS-2 is clearly related to SARS-1. So, yeah, there can be a SARS-3. There might be something else in between. It could be the Australian. No, it won't be the Australian. Could be the, well, the Middle East is interesting because we didn't know camels harbored these coronaviruses. So anywhere there's a coronavirus, and they're all over the place. It could be the South American Respiratory Syndrome coronavirus. SARSC, something like that. You got it, wherever it comes from. What is the actual length of time for actual immunity that matters? The cardiologists and HOTAs now down to 1 to 2 months, and they have a new answer every 2 hours. So cardiologists shouldn't be talking about this because they don't know. And HOTAS does know better, but he's out of control. When you get a booster, you have a boost in antibodies and T cells that, so the levels are very high for up to 6 months, and then they contract, and now you have memory, which is perfect because that's how the immune system works. You don't have an immune system keeping high antibody and T cell levels all the time, so they go down. But then when you're infected, you have a memory recall that prevents severe disease. It doesn't block infection because, as I said earlier, the memory response is a bit delayed. And so they don't get this. So what they're trying to do is actually prevent infection, which is impossible. You're not going to do that because the antibodies are always going to go down after 6 months. You're not going to prevent infection. It's just not a thing to give a boost every 6 months. Makes no sense whatsoever. So don't listen to the cardiologist. You're absolutely right. The YouTube channel Debunk the Funk. I think we had that as a pick once. It's doing a chapter debunking review on Kennedy's anti-vax book. He constantly mentions and promotes Twitter. That's great. We should promote them then, because this cross-promotion is so rare in science social media, right? Everybody wants their own little empire, and they don't cross-promote because they think they're going to lose a chunk of their empire if they do that. It's really too bad, but glad they do it. We'll have to have them on, right? That would be cool. Vincent Price's version of I Am Legend is, I only saw 5 minutes of it. It is a scene where Price, this is about a cancer curing virus that goes awry and kills people, right? And the lady is sitting at the table. She has the 60s hairdo. My mom used to have, where it grows up, it curls around, and then curls under, right? It's all straight. She says, and she's very smart. She says, "Are you sure it's more transmissible? I couldn't believe this. This is 1960s. And he said, and this is why it's Vincent, he said, "I very much doubt it." Yeah, he's channeling Earth's virology professor. I hate to bring this up because I'm not a fan of the JC. The Pfizer docs claims the risks may not outweigh the benefits. Well, he's just full of it. He doesn't know what he's talking about. Please stop listening to him. Please. You're getting all kinds of misinformation. Of course, the benefits outweigh the risks, not the other way around, of the vaccine. I haven't read the docs. I'm sorry. Would you like me to? I can do that, but I don't know when I'm going to get to it because the next week is really a mess. The day the Earth stood still fits our current situation well, and this old dude recommended it. Okay, recommendation taken. I will read it. Excellent. What are your thoughts on the fact that Congress removed $15 billion from the omnibus spending bill that was designated for COVID testing, treatment, and vaccination in the U.S. and in poor countries? I think it's highly unfortunate, and it is a political move, of course. It's not an intelligent move. And it's really unfortunate. It should go to poor countries, right? We should be helping them. And they have never liked to do that, only when it becomes a threat here. It's been a threat. Now they see the threat is lifting. So I think that's unfortunate. We should be spending this on helping others for sure. Thank you, 8440, for your contributions. I have the list of virologists from ASV that I reached out to in 2020. Do you think it's appropriate to approach them regarding career select suggestions, like non-COVID matter? I don't know what you mean. Not sure what you mean. So you reached out to these people. Now you want to reach out again for career advice. I don't see why not, no matter what it has, as long as it has to do with virology, right? I think that would be fine. For sure. What's the video software you're using for conferencing? What are you talking about? Like here, now, or on Twiv? This one? This is called Ecamm. ECAMM. You can just search for it. The one I use for Twiv is different, but I don't think that's what you mean. Daniel's Twitter account. Just go onto twitter.com and search for Daniel Griffin. It's Daniel Griffin, MD. Here, let's do it. It's really easy to do this. Twitter.com. I'm going to share my screen. Hey, producer, share my screen. Thank you, producer. So here is, so yeah, my Twitter header. It's Earth's Virology Professor. And so I want to search Twitter here in the upper right. Daniel, if I start typing, Griffin. Here he is. Daniel Griffin, MD. At Daniel Griffin, MD. There you go. That's his, that's the way to do it. Just go to twitter.com and go to search Twitter. And just be careful because sometimes there are imposters, right? But I know this is Daniel. It's quite obvious, right? Say, producer, turn off the screen share. Thank you. Let's see. Yeah, I'm still wondering what was the Vincent guy, virologist for West Nile, but I can't remember his name. Are there any inhaler antivirals? The one I know of is the antiviral, the flu antiviral. So we have Tamiflu, which is the pill, and Relenza, which is the inhaled antiviral. Now, of course, there is an inhaled monoclonal for respiratory syncytial virus, which is different, given to infants. But that's what I know. Okay, yes, aromaticus. Older people are immunosenescent. That's absolutely right. Not really suppressed. Suppressed we should reserve for immunosuppression, say, caused by virus infection, caused by treatment with steroids or similar compounds or other things for transplants where we purposefully suppress. But yes, you're absolutely right. Immunosenescent. See? Yes, very good. Excellent. And I know that someone gave Charlotte her Daniel's Twitter already. I can't see that ahead of time, so I answered it. But I also think it's good to teach people how to find it rather than give them the answer, right? The mark of a teacher. Is the IQ of humanity likely to drop due to COVID-based brain shrinkage? No. And if Amy were here, you know what she would say. It can't drop any further. That's not nice. I understand that. She may not have said that. I don't think so, no. Not enough people are having brain shrinkage to do that. So hypoxia is when you cut off oxygen, you reduce oxygen, which happens certainly in COVID, right? So a number of months ago, we had a neurologist from Columbia on Twiv, Kieran Thacker. They did a study, which we talked about, where they found brain damage in COVID's patients. No evidence for virus reproduction in the brain. And I asked her, so what causes the damage? And she said, most likely hypoxia and inflammation, the cytokines that are produced in your lung in response to infection, which of course get in your blood and they circulate everywhere and they get in your brain. Yeah. Why does infection make people's hair fall out? We discussed this a couple of Wednesdays ago. Since the stress causes hair loss, and it's most likely the stress induced by infection. Yep. And you know what, folks? 615 people tonight. I'm so impressed that you guys come back every week. Hit the like button. We have 300 likes. Can we hit 500 tonight? You've recommended books and movies about viruses. Any documentaries you would suggest? I don't have any at the tip of my tongue, but I'm sure our audience here will come up with some, and they probably have already come up with some. There are several good ones, but I just don't recall offhand. This is a good philosophy. Life is for living. Yeah, don't be afraid. I don't think you should be afraid. Yeah. Yeah, sure, I'll cut your grass. I have time to cut your grass, and I'd be doing less teaching. What do you think is more important? Yeah. Yeah, don't be scared about chronic wasting disease. I'm not scared. I'm just fascinated by it, that how it works and so forth. Yeah, I'm really fascinated. So, by the way, while I'm looking for another... Yeah, that's right. I don't live in Arizona. We have winter and summer grass. Here, I'm in New Jersey, where in the winter the grass doesn't grow. Absolutely right. So my travel plans are shaping up for the year. Going to Zurich in May. Going to Nebraska, Omaha in May. Going to Madison, Wisconsin in July. Perhaps Washington, D.C. in June for the ASM conference. And what else? September looks like we have Manchester, UK on the agenda, and perhaps October, Honolulu for an immunology conference. I don't really want to go all the way to Hawaii, but it's to do a podcast, So that if you're any of those places, let us know and we can meet up. Can you list in bullet form the things we were wrong about COVID? Yeah, I'm gonna have Steve Morse on TWIV in the coming weeks, and this is his thing. He gives talks now about what we got wrong, and I'd like him to do that. That's a good point. Yeah, absolutely. Thank you, Fluid Systems, for your contribution. With virus out of control in South Korea, are you concerned the Omicron sub could cause mutation in new global wave? No, so we could have further immune evasion, that's for sure. So Omicron could change and evade antibodies even more. But in all the evasion of antibodies, the protection against disease, severe disease, remains. So I'm not concerned, no. So here's the thing. This is a similar situation for influenza virus. That virus every year changes, every year or two, and the variants evade antibody, and that's why they become more fit and they spread. They transmit among people very well because they're immune evasive, like Omicron is. And every so often, the immune evasion is sufficient to cause an increase in severe influenza. So they keep an eye on that. And for flu, they know that when antibodies get to a certain level, then they should change the vaccine because that signals more disease. We don't know that for SARS-CoV-2 yet because, Omicron is not neutralized at all by some people's sera, yet they're still protected against severe disease. So we're missing something. We need to know what the correlation is. But that's the only case where it could change and now evade antibodies sufficiently to cause more severe disease, but it hasn't happened yet. I'm not worried about it, though, because I think we can deal with it. We just change the vaccine, right? Since viruses are largely RNA and DNA strings, how influential were viruses in the general mutations of life on Earth? So I would say that viruses have been influential in gene exchange among organisms. I don't think... so they have played some role in mutation because they can... some viruses can insert into genomes and so forth. But I think that's minimal compared to their ability to pick up a gene from one host and move it to another. We know that that has played a huge role in evolution of bacteria, so bacterial viruses do that, and it's likely to have happened with eukaryotic viruses as well. So the one exception are the retroviruses, which integrate a DNA copy of their genome into the host cell. And our genome is 8% retroviral from all the infections we've had over the years that have left behind these traces. And these sequences can move around, so they can impact the genome as well. They can have mutagenic effects for sure. But I think that's less than the effect caused by gene movement. But it's a good question. I like that as well. So Spamlet is talking about stink bugs. Yeah, so the powerful smell. I don't swat them, but did you know that stink bugs, they have a symbiont in them that helps them, that provides them with molecules that they need. And the paper we did on Twevo showed that you could replace it with E. coli. A one amino acid change in E. coli allows it to become a symbiont of a certain kind of stink bug. It's so cool. It is really cool. Ay, ay, ay. Where does Amy get her sense of humor? Well, that's Amy. You know, it is the way it's her. That's how everyone is, right? Most people don't work to get a certain property, I guess. It's just the way she is. We need a TWIV on ruminant viruses. Okay, we could do that. You ask, you say jump, we say how high. Yes. Is an N95 mask enough protection in the gym if you're over age 65 boosted where masks are not required? If you would like to wear a mask, yes, a fitted N95. Make sure it's been fitted so that it fits you properly. Then that would be absolutely, yes, absolutely. 384 likes. Come on, folks. And a lot of people left after I asked to like. Okay. Thank you, Doreen, for your contribution. Looks like a lot of people are leaving. Oh, they're just saying goodnight to Amy. Thank you, Maytree, for your contribution. Okay, now that mRNA vaccines have proven to be safe and effective, is the technology settled so that drug trials need not concern themselves with the platform? Well, technology is never settled. They can always try and modify it, right? But, you know, depending on, you're going to still have to do a phase 1 safety, right? Because whatever you put in it, whatever mRNA you put in it, even if you use the same composition of the nanoparticle, you're going to have to make sure that it doesn't have side effects. So, no, I don't think it's going to be reduced at all. Have you any updated info? So they just requested emergency use, right? Yesterday. So when would the FDA approve it? They could do it within a week. It could be very quick. We'll see. I have no inside information. Maybe Daniel will talk about it tomorrow. So the virus... Wait, thanks for your streams. Educational, you always try to answer all questions. I do. I go through the list. We are 3/4 of the way down tonight. We still have 22 minutes left, so maybe I'll get through them. I like to teach. I feel I have something to say. I have a 40-year experience with viruses that is of some value, so I try and impart it. Now, yes, not everyone does it. Not all scientists can, because not all scientists can communicate well. Not all scientists want to, and not all scientists have the time to do it. I made the decision at some point that I wanted to do it, and it has consumed more and more of my time. I feel that the reward has been worth it, so I continue to do it. So the virus doesn't get weaker, rather the population doesn't get the same level of disease because they have changed. Yes, that's one idea, absolutely. The virus doesn't have to get weaker, no, because I think immunity makes a big difference. That's right. Daniel says, never miss an opportunity to test. No, I don't agree with that. Every time he says it, I keep my mouth shut. No, we shouldn't test everyone. You're not going to learn anything by testing everyone. No, if you want to do a study on how influenza circulates in every person in the U.S. over the winter, that would be a dumb experiment because you never need to test everyone. You never take serum from everyone. You take it from a fraction and you extrapolate. Never pass up an opportunity. Never miss an opportunity to test. I don't think that we are in the beginning of the outbreak. Yes, not anymore. Not anymore. If you are double vaccinated and boosted, if you test positive 7 days after you first test, are you still shedding? You might. You might. It depends how much. I don't know how much you would be. So this study hasn't been appropriately done, right? That's a really good question. If you're fully vaccinated, how much infectious virus do you shed and for how long? And you might find one study on this that's looked at infectious virus. Pathetically lacking. And all the work that we have done, people just use PCR and it doesn't answer the question. So I wish I knew the answer. I suspect it's a shorter shedding and lower titer and probably not enough to infect people on the whole, but we just don't know. If I visit New York, can I visit the incubator? You bet. Yeah, of course. You can send me an email, Vincent at microbe.tv, and we'll, as schedule permits. Sometimes people say the day before, hey, I'm in town tomorrow. Can I visit? No, the whole day is booked. I'm sorry. I have to do work here. So give us some time. And you're welcome, plumber. I'm glad you came because I know you could be plumbing and making a lot of money. So I'm glad you came and learned something. How to return to normality, when you have a 1-year-old, aside from testing all visitors that come? Well, no, you have to get vaccinated, I think. But you can mask if you have visitors. The visitors are presumably vaccinated. But if you're worried that they might be shedding, which is a valid worry, yes, you could test them. Or if it's a lot of trouble, and maybe it's not so essential, right? And you can wait a bit longer. The sign said free testing. I got the test and got a bill. Of course. Don't ever believe free anything. So in New York, they're right outside the incubator. There's always on the corner a testing kiosk. Free. No cost to you. I don't believe it. Not that I want to be tested. Why would I? I have no symptoms. I'm not going to be tested. If I had symptoms, I would get tested to see if I needed, but I have no comorbidities. I wouldn't take, I don't need to get tested. With immune evasion, would not an update periodically now for Omicron make sense? No, you will never get past those breakthroughs that end up in the hospital. No, unless you boost every 6 months. And the Omicron-specific vaccine tests that have been done didn't show any improvement. So no, I don't think it makes sense. I think we're doing the best we can do at this point with these kinds of vaccines. Yes, influenza doesn't cause long COVID for 2+ years. What is the point? You want to be tested to know if you're going to have long COVID, but you can't do anything about it, so I don't understand what you're saying there. Please clarify. I will read the Pfizer documents, okay, because I think there's a lot of brouhaha over nothing, so I will have to bite the bullet and read them. I'll do it, okay? Give me a couple of weeks. What is your take on SARS-CoV-2 and RATG13? Totally different viruses. One did not come from the other. The RATG13 was identified in 2013. His genome is 1,200 bases from SARS-CoV-2. It's not the ancestor. There are other viruses actually in Laos that are closer to SARS-CoV-2. Prion diseases can't be passed human to human. Yeah, they can be. Yeah, cannibalism, obviously. So the Kuru was a disease of the Four people in New Guinea, was passed by cannibalism. But many cases of Creutzfeldt-Jakob were passed by corneal implants. We used to make hormones purify them from people, and they were contaminated with prions. Surgical procedures. You do surgery on a patient with a prion disease, you don't know it. You autoclave your instruments, but they don't destroy the prion, and then you pass it to another person. So, yeah, it can go from person to person, right? Eventually, when we grow our meat in labs, many risks will be mitigated, and it will be humane. Yeah, I agree with you. I love the artificial beef, right? Impossible, etc. I love it. The taste for me is fine. The idea is great, but I think there are going to be a lot of people that won't go for it, and there'll still be a market for processing animals, unfortunately. Yeah. What would a live cold attenuated COVID vaccine look like? Well, the flu mist is a cold adapted infectious attenuated vaccine. In other words, the virus can only reproduce in the upper tract. It cannot go down into the lungs. And so one could make a virus that could do that. And in theory, it would be also attenuated. It would cause less disease. But we don't have that. But that's the idea. And we have one in flu mist, so you can follow that parallel. Flu mist, it's sprayed into your nose. It's an infectious attenuated flu vaccine, attenuated, and it's cold adapted. It just reproduces in your upper tract and gives you immunity. Unfortunately, still not good enough, in my opinion. We can do better than even flu mist. Chronic wasting disease has been known for 3 decades. Yeah, that's not forever, is it, Doug, right? It's just 30 years is a blink of the eye. It's not even as long as I've been doing virology. So it was recently, I remember the day it was discovered, okay? That's why I say it's a new disease. It's all relative in how you say these things. That's correct. Here's the malaria vaccine, Mosquirix, which we covered on TWIP. It is going to help. It's not perfect vaccine. It doesn't prevent as much as we'd like it to, but it's good. It's a good start. Absolutely. Thank you, Ian, for your contribution. Incidence of MS increases towards the poles. I have a theory that it may be due to us meeting many, many viruses, not parasites or bacteria, near the equator. I don't know why that would prevent MS, because MS is an autoimmune disease. The antibodies against the virus will attack your cells. So I'm not sure why having other virus infections would do that, unless they're interfering with it. Is that what you're thinking? I don't know. There are lessons to be learned from our mistakes with COVID, but will we learn them? No. I agree, Tom. We are not going to learn them. We have a short memory. It's generational. It will be forgotten, and the next generation will not do it. They'll make the same mistakes, unfortunately. Can you explain how a post-infectious ileus process happens, how long it tends to last? Could GI dysmotility possibly last forever? So these are COVID-induced GI issues that you're talking about. We don't know. So it could be the consequence of inflammation, right? It could be a consequence of antibodies against the virus that are cross-reacting with GI tissue. And we'll get the answers eventually. I'm sure we will understand the basis for it, but will it last forever? I doubt it. I think at some point it'll be treatable, because if we say, oh, it's because of these antibodies, we can fix that. We can interfere with that. So I don't think it would last forever, no. As even mild infection can result in long COVID, why wouldn't I try to avoid any infection till there's many therapeutics or global pandemics over? Well, you could if you would like to. in my opinion, the numbers say that vaccination decreases the frequency of long COVID substantially. And that works for me. I want to live a normal life. And there are lots of infectious agents out there, and you can't be afraid of them. I think at this time we have the best we can do. But if you're afraid and you don't want that low frequency of COVID that could result even if you're vaccinated, then by all means, avoid infection. I just tell you what I've decided to do based on the data that I've seen. I've evaluated the risks, and I've decided that they are worth taking. Spanish flu is much worse, many more infections, many more people died for a variety of reasons. But we recovered from it, and we'll recover from this one. We have more from this one, right? All three of my kids have had COVID. Our pediatrician doesn't feel the vaccine is necessary, but Disney Cruise Line requires it. Yeah, well, you have to make a decision as parents of your children, and I can understand if they have had COVID, your position, but yeah, don't go on the cruise. You can go another time, right? Do I believe BA2 is significantly more contagious than Delta or prior variants? I don't know about BA2 because the studies haven't come out that are definitive. So we just did a very nice study on TWIV. We reviewed it. It's dropping tonight on BA1, and that's not been done with BA2. BA2 is not completely displaced. So BA1 displaced Delta almost overnight, right? And BA2 isn't doing that. It's making inroads, but it's much slower. So it's not as big a change as Omicron. It's probably an incremental step or an incremental change. So, yeah, I think it could very well be that the increased transmission or the fitness of BA2 is a consequence of immune evasion over BA1. We have to just wait and see that. I just don't know. Don't you think by unmasking before everybody's vaccinated, we run the risk of the virus going either way, like increasingly evasive? Well, you're never going to get everyone vaccinated, right? Because there's some people who will not. And they're the only population that I think, well, there's the immunosuppressed, immunosenescent, and the young kids who haven't yet had the opportunity to be vaccinated. But right now, in New York City, the level of infection is very low. It's in the so-called green level for CDC. So there's no reason. There's very little virus circulation as far as we can tell from cases. So I don't think that's an issue. No, I don't think the virus will become increasingly evasive. No, there are enough people not wearing masks anyway. It's not going to stop infection, right? It's going to protect you. But in the overall population, it's not going to change the penetrance of the virus, right? You've said elsewhere, that was on Lex Friedman's podcast, that you don't think life exists elsewhere in the universe. Why do you think this? I just think it was a random event that happened here, incredibly fortuitous, and it's not likely to happen anywhere else. That's all. Viruses were not around before cells, but self-replicating RNAs were, which then became viruses. And I just think that was, if you go through all the things that had to happen to get to that point, I just don't think it's happening anywhere else. Because it is such a series of rare events that have to happen. And I understand the universe is huge and there are many opportunities. And maybe I just don't grasp its hugeness, but I don't think it's anywhere else. I could be wrong. It's fine. How many universities have standalone virology departments? Very few. I would say less than a dozen in the U.S., and usually it's part of microbiology. So you get a degree in microbiology, yeah. Absolutely. Vaccine issues aside, why is dengue often more dangerous the second time you get it? So when you get, there are 4 serotypes of dengue. You get one and you get immunity to that. But when you get, say, infected with a second serotype, the antibodies you make are against the first serotype. They bind to the second serotype. They do not neutralize its infectivity. And now that virus with the antibody on it could get into, say, macrophages, and you get more virus produced, and you have a lot of cytokine production as a consequence. So you get this abnormally lethal disease with the second infection. If viruses want to keep their hosts alive, why would they be deadly or harmful? So viruses have no desires. They have no will. They have no cognition. They have no sentience. They're just chemical reactions. So they're not making any decisions. The reason they're deadly is mostly because of our immune response to them. In COVID, that's where the serious disease comes from, our immune response causing damage. It's an overabundant or overexuberant immune response to the virus. So most viruses, in fact, do not damage their hosts. They coexist with them quite well. It's only when a virus changes hosts that seems to be a problem. Okie dokie. Producer, yes. I guess I would still like to have a producer to work with me, but that would require someone at the incubator. All right, we are now 4 minutes away from... I mentioned I was using a new conferencing software. I don't remember. So this eCam I've used on this live stream from the beginning. At the beginning of the pandemic, we used Zoom for TWIV, but Zoom sucks big time for podcasts. The video and the audio quality is crap. Although, if you go back to my early COVID TWIVs, I was able to wring good quality out of Zoom. It was not worth the effort. And the problem with Zoom is that my audio is on one channel, but everyone else is on another one. So when Dixon coughs, screws everything up. So I found Zencaster, where everyone's audio and video are recorded on separate channels, and then I edit them in the end to produce that lovely production where everybody looks great and sounds great. Yeah, but I'd love to teach someone else how to do that. We Heard the Bells is a very good documentary on 1918 flu. Okay. Excellent. BBC had a series on Discovery called Patient Zero. There you go. Okay, I haven't seen any of these, but glad for the recommendations. A new live TWIV. Vincent teaches while cutting people's grass in live streaming. Sounds good. Sounds good. You were born in Manchester. I like Manchester. I've been there before. And you know what? They have really good Italian restaurants in Manchester. So I'm looking forward to going back. Okay, so let's get through here to the end. I didn't get through all the questions, but come back next week and bring your questions back. What a nice community, folks. I'm looking for more contributions just to thank everyone. And yes, I will read the Pfizer documents for sure. Given that viruses are not alive, shouldn't virology be a branch of chemistry rather than biology? And viruses no longer considered microbes. Well, the problem is that to study viruses... So the early people who studied viruses were chemists. So Wendell Stanley, who crystallized tobacco mosaic virus, was a chemist. And he bollocks the whole thing up. He said... it's the protein that's infectious for this virus. He was wrong, and he should have taken away his Nobel Prize. Anyway, the reason it's not just chemistry is because you need living cells to study their reproduction. So that makes it. And microbiology, all right, are they a microbe? It's something you need. You can't see them in the microscope, right? That's what microbiology is all about. But historically, they've been just put in microbiology departments. But I do think it's a science unto its own, because it is a little chemistry if you want to study the structure of viruses, for example, but it's also cell biology and molecular biology, genetics, etc. So it's a unique science, and it really should be on its own, but it benefits from cross-feeding from other disciplines for sure. Vincent, and the incubator is the best thing YouTube has. I don't think I'm the best thing YouTube has. There's so many great channels on YouTube, but I'm glad you find it useful, and I hope to continue to be useful. I have my little corner that we inhabit here, and there's been this 2 or 3 year period when we've been useful, and hopefully we'll be continue to be. I thought Daniel said chances of long COVID. I don't think those numbers are right, Caroline, because they do those surveys with questionnaires, and that's not how you diagnose a syndrome with a questionnaire. It's just not good enough. I think it's lower than that, in fact. Knowledge is temporary, but greed is forever. Greed is a big cause of why we're not prepared and why we took so long to respond. Absolutely right. Check out my virology blog post from early 2020. This pandemic could have been prevented. In fact, let's see if you search for that. This pandemic could have been prevented. Let's see if we get my post. You crappy Google. Here it is. It's on the first page, actually. Let me show you. Hey, producer, turn on the screen share. Thank you. So I searched this pandemic could have been prevented. There's the first page. And there it is, right? Right there. There it is. The 30th April 2020. Could have been prevented. Read it. It's good. Nobody cared. Nobody cared. Turn off the screen share, producer. Let's get back to wrapping up here. And actually, this is a good way to put it. Viruses are evolving entities that are intimately related to cellular life. They're parasites, and everything that happens in an infected cell. Yeah, but it's a good idea, and it helps to understand. Ditching says, might they become a yearly vaccine? I don't agree. I don't think we need it, but I have covered this extensively in this stream. It could be that, like flu, the disease severity increases at some point, but we don't know what change would cause that because it hasn't happened yet. I'm open to seeing it, but so far it hasn't happened, and so far we just need this one vaccine for sure. All right, folks. Thank you all for coming here tonight. I appreciate your participation. Our moderators, Les, Steph, Tom, and Vanity Nutrition. Thank you all for being here and doing your wonderful job. And yes, TWIV has been around since 2008. We were audio only until the pandemic. So there's a silver lining in that cloud. We are now video. Come back next week, folks, for your questions. You all have great questions. I appreciate being here with you chatting. In the meantime, be safe and have a great week. Thank you and good night.